Here we go. Welcome everyone to this session, and thank you for joining us today. The live transcript is available if required. If you prefer to hide it, just click the live transcript button and select hide subtitles. The session will be recorded and the link will be sent to you afterward. It will be available to view for 7 days. Please use the Q&A function if you have any questions that you would like Professor Jose Luis Puente to answer at the end. Feel free to use the chat to introduce yourself and for general comments. Allow me to introduce our speaker, none other than Professor Puente, who is also Associate Senior Editor for the Journal of Medical Microbiology. He is a professor of molecular microbiology at the Instituto de Biotecnología of the Universidad Nacional Autónoma de México, in short, UNAM. He received his bachelor's and PhD from UNAM, and he followed it by postdoctoral training in the microbiology and immunology at Stanford University under Dr. Gary Skulnick. He has been a visiting professor in Brett Finlay's lab in the Michael Smith Laboratories at the University of British Columbia. He was raised in a family of non-scientists as his parents, like many of us are, who did not have the opportunity have a university degree due to the need to work from a very young age. His parents, however, instilled in him commitment, hard work, sense of gratitude, and generosity, and fostered the importance of having a loving and united family. Thus, his path to becoming an independent scientist has been paved by his family's unconditional support and love, who continue to inspire him. He also recognizes that his journey would not have been possible without the trust and support of his mentors, colleagues, students whom he had the privilege to work with. Along the way, he had the opportunity to meet great scientists who are now friends and collaborators. Jose and I had the opportunity to meet at one of the Cold Spring Harbor host pathogen meeting almost 20 years ago, when both of us were just starting as faculty members. And that friendship continued to be a collaboration beyond science. And I'm really pleased to have Dr. Jose Luis Puente as the speaker of the day, and I welcome him to the podium.
Thank you, Kalai, for the nice introduction and for having me today to speak in this seminar series. And welcome everyone. Good morning, good afternoon, good evening, wherever you are. Let me try to get this to work. So as you can see, the title of the talk, I'm going to be talking about pathogens, but a different aspect of most of the talks that we have had so far. So I'm going to be talking about gene regulation. And just before I start, I invite everyone to consider the Microbiology Society journals for your publications. So take a look at the website to see the advantages of publishing in the Microbiology Society journals. And just a quick note of where I am. As Kalai was mentioning, I work for the Instituto de Biotecnología of the Universidad Nacional Autónoma de México. And we are located in, not in the main campus, which is in Mexico City, but in a smaller campus located in the city of Cuernavaca, Morelos, which is, we're pretty much at the center of the country and south Mexico City, so close enough, but also far enough. So it's a convenient place to work and in a beautiful setting. And as you can see in the picture, this is one of the buildings of the Institute after a rainy day, we got a couple of rainbows, so come to visit if you wish. You're welcome to visit the Institute. Just a brief outline of my talk. So I'm gonna be, the subject of my talk are actually fascinating pathogens, as all bacteria are. In this particular case, I'm gonna briefly introducing what the attaching and effacing pathogens are, and also briefly introducing the hallmark of this family of pathogens, which is the locus of enterocyte effacement. And then I'm gonna move on to talk about global regulation, and in particular, most of the talk, I'm gonna be mentioning some insights about the function of some of the lean coded regulators, which are the subject of my talk. And at the end, I will summarize with current knowledge and current trends and some conclusions. So the attaching and effacing family of pathogens is integrated by, in particular, 4 pathogens, 2 pathotypes of E. coli, and which are important human pathogens. One of them is enteropathogenic E. coli, also known as EPEC, which is the cause of acute diarrhea in young children, and is still a public health concern in low-income countries. And also enterohemorrhagic E. coli, which came to the scene around 1982 when it caused the first big outbreak in the US and caused the death of several people. And this is also a relevant pathogen because it's still a major problem in some places, and a major cause of hemolytic, the hemolytic uremic syndrome, which can be deadly, and then the relevance of EHEC. And also Escherichia alberti has come to the scene in recent years, also as a cause of gut watery diarrhea in children and adults, and it's been recognized also as an important pathogen, and you will see it shares some of the virulence factors that characterize EPEC and EHEC. And the other organism, which is also very interesting, is Citrobacter rodentium, which is the causative agent of transmissible murine colonic hyperplasia. And the advantage of having Citrobacter rodentium as a member of this family is that it has provided an animal model to understand different aspects of the colonization and the infection process of this family of pathogens. So it's been an important and very relevant surrogate model to understand EPEC and EHEC pathogenesis. This figure illustrates some of the several aspects that are currently known about the virulence and the pathogenesis of EPEC and EHEC, mostly based on Citrobacter rodentium studies, which depicts the function of different effectors that these bacteria produce and inject to the epithelial cells to cause, among other things, diarrhea, and in some cases they subvert different signal transduction pathways and different functions of the cell in favor of the bacteria, which take advantage of this to establish an infection on the surface of epithelial cells. So in general, these pathogens are not intracellular pathogens. They stay attached intimately to the cell surface, as you can see in this cartoon. And the process by which these bacteria actually establish this intimate attachment is a fascinating process, which I hope we have the opportunity to talk about in another time, because the whole thing is very interesting but complex at the same time. So as I was mentioning, the hallmark of this family of pathogens, of what characterizes and identifies these pathogens as a family, is because they share a pathogenicity island that we know as a locus of enterocide effacement. And the name comes because it was named that way because it's responsible for these lesions that these pathogens cause on the surface of epithelial cells, that is called the attaching effacing lesion, which is characterized by the effacement of the microvilli and then the loss of water and diarrhea, etc. But in terms of the contents of this pathogenicity island, which is composed by 41 genes that are organized in 7 operons, is that it encodes different virulence factors that allow these bacteria to colonize or establish the colonization and this intimate attachment to the surface of epithelial cells. And one of them is a type 3 secretion system, which is also known as an injectisome, which is literally a molecular syringe that injects bacterial proteins inside the epithelial cells, and these different proteins that we know as effectors have different functions, and as I was mentioning, the capacity to subvert different signal transduction pathways and different functions on the cell, including this ability that they have to reorganize the actin to form these pedestals where the bacteria sit to establish the colonization. Along the genes that have to do with biogenesis of the type 2 secretion system, there are also translocator and effector proteins. The translocator proteins have to do with the injection itself of different effector proteins, and the effector proteins have those that are gonna have different functions inside the cell. And there are also chaperones that stabilize the different effectors to bring them to the base of the type 2 excretion system to be secreted, and regulatory proteins, which are going to be the main subject of my talk today. Despite the fact that A pathogens share this pathogenicity island, you may imagine that within each pathotype there are also diversity, and between them there are also differences that characterize the disease they cause. In the case of EPEC, as I was saying, there's basically they cause the acute diarrhea in children in general under 2 years, and but there is also EHEC that, in addition to the lead pathogenicity island, they also contain a phage that encodes a Shiga-like toxin, which is the cause of the uremic syndrome. And as I was saying, it can be deadly, and those the risk of having an infection by EHEC, which is in general acquired from contaminated food. But it's still a major problem even in industrialized countries, and there's still focus of attention by different investigators. So in general, in terms of studies in vitro, and considering that EPEC is less dangerous in terms of working with it, it has been also one of the preferred models to understand or to study the function and the regulation of virulence factors together with Citrobacter rodentium, that provides an animal model for different studies. And one of the interesting characteristics when you look at the difference between these pathotypes is that, in addition, for example, in the case of EPEC to the pathogenicity island, it also contains a large plasmid that encodes a very characteristic pilus that is known as the bundle-forming pilus, which is responsible for also a very characteristic a phenotype of EPEC that is known as a localized adherence. So basically, BFP allows EPEC to form bacterial aggregates that are very efficient at binding to the surface of the epithelial cells. So a few of the cells that actually make direct contact with the surface can start injecting through the titration secretion system effectors and then establish this intimate attachment to the surface that is actually illustrated in this slide. So we take EPEC as a model, and we go through the stages that EPEC transit to establish an infection. As I was mentioning, the BFP, the bundle-forming pilus, allows the bacteria to form these aggregates that establish the initial adherence to the microvilli, and then the titration secretion system starts injecting effectors, in particular one fascinating protein that is called TIR, that is going to become the receptor of the bacteria that interacts with another membrane protein called intimin, and that's the way they establish the intimate attachment. But at the same time, they keep injecting other effectors that are going to cause different changes in the cell until the intimate attachment is established. One of the interesting features of EPEC is that in this plasmid that we know as the E. coli adherence factor plasmid, because it was known even before the pathogenicity island, the Li was characterized, that it was responsible for this localized adherence phenotype, which is very important for the infectious process. And actually, in the old days when volunteers studies were still valid, it was demonstrated that actually BFP is an essential virulence factor to even establish an infection in adults. But the other interesting thing is that this particular FIMB is regulated by a cluster of 3 genes, an operon of 3 genes that is called PER, where an ARA-C regulatory protein or activator controls the expression of the bundle-forming pilus, but also controls its own regulation, and then the expression of a third protein that I'm going to show later that has to do with the activation of the pathogenicity island. So keep this in mind, because it's one of the interesting characteristics of EPEC. But this illustration is just a cartoon of this very complex but also fascinating structure that we know as the injectisome, because it's literally a molecular syringe. Don't panic with this slide, but the idea of showing this is one of the figures of the models of the regulatory cascades that are involved in the regulation of this pathogenicity island. And the idea of showing this slide is just to illustrate how complex, as we know more, as we understand more, how complex this network is becoming. And I'm not going to go into the details of this, but it's just something that I want you to keep in mind is that most of these regulatory elements that influence the expression of the Li actually target one particular promoter, which is the promoter of this first operon that we call the Li1, which encodes in the first gene a protein that we know as LER. And I'm going to be talking about this particular protein. So just keep in mind that most of these regulation based on all these different factors channel towards LER. And this is trying to bring down to a summary what I was showing in the previous slide. And one of the interesting characteristics when we look at the regulation mechanisms of this pathogenicity island, and as I was mentioning, basically most of the control is on LER expression, is that it's influenced by different regulatory proteins that are either global or ancestral regulators. So genes or proteins that already existed in the ancestor of IPEC and IHEC when they acquired the pathogen ST island, and that were just integrated into the regulation of the LE. But there are also genes that are encoded in the accessory genome. In the case of IPEC, I was mentioning per-C, and there are orthologs of per-C encoded in also in the accessory genome of IHEC. But there are also other regulatory proteins encoded in either other pathogen ST islands or phages, as well as small RNA. So every time is more and more is known about the role that small RNAs play in the regulation of these complex pathogen ST island. And once again, most of them targeting the regulation of LAIR, which is the master regulator of the island. And I'm going to be focusing mainly in the role that LE-encoded regulators play in the regulation of this pathogen ST island, because they're not only the more interesting ones, but they're the ones that actually arrive or were acquired by horizontal transfer events together with the rest of the pathogen ST island. So it's perhaps not too speculative to consider that these regulatory proteins were involved in controlling the expression of this set of genes, of the LE in the ancestor without the need of other regulators. But it's not too speculative, but it's still speculation because we don't know. So the idea of this slide is just to summarize what is known and what was shown in the previous slide, and let you know that I'm going to be focusing on those regulators that are encoded within the pathogen ST island. So before I go into the details, I just would like to show you this slide, which is a nice model of what is considered happens during the transit in a new host, or what happens during the host. But the idea is to give this message that when we study gene regulation given bacteria, so we try to control the conditions we test in the laboratory, and we are looking at the particular window and situation where we are assessing a particular signal or a particular stress, and how that influences the expression of the genes of interest. But when a bacterium is actually colonizing or initiating the infectious process in a host, is going to confront many different signals. Some of them are going to be signals that are an indication that the bacteria should turn out the virulence genes because they're not going to be needed, and some others are going to be actually signals that are going to be inducing the expression of virulence factors. So the thing is that when the bacterium is going through the intestinal tract, is going to be facing big changes along the way, and the way they respond is by basically generating a consensus response based on all these signals. So we need to keep that in mind when we study gene regulation in vitro, because even when it might be very informative and can give us an idea or elements to at least hypothesize of what might be happening in vivo, what is happening in vivo is not nearly related to the conditions that we can simulate or try to mimic in vitro. So this slide or this cartoon also shows some of the knowledge that is being generated using even the Citrobacter rodentium mouse model, and that is also giving us an idea of what might be happening in vivo. But still, we cannot control everything. We cannot modify everything, because the combinations that we can generate, either mostly in vivo, are just too many to be able to really try to have like the big picture of what might be happening if we include all these variables that seem to influence the expression of VMS factors. So, but still, we so far, as I was showing also in this complex network of regulatory elements, that not only us, but many different groups that have been interested in the study of gene regulation or the regulation mechanisms in this group of bacteria. But we have learned many things, and more components and elements are coming into these models of the regulatory network, and it only reflects that we still have a lot to know. So also the idea of this slide is just to set the conditions or that we use in vitro to simulate what we call repression conditions and what we call activation conditions. And fortunately for those that are interested in studying these organisms, this medium DMEM, the Dulbeckus modified Eagles medium, that is used to actually grow monolayers of cell lines, is actually a condition that IPEC likes to induce the expression of its virulence factor. So it's a very convenient condition, but it also works to study the localized adherence or the formation of pedestals on epithelial cells. And also by luck, growth in rich medium represents a condition where this pathogen ST island, the locus of enterocyte effacement, is in general downregulated. So we are lucky to have these laboratory conditions where we can have both ends of the activation or repression of the pathogen ST island, and it's been an advantage also to understand the role of different regulatory proteins. So if you see a yellow flask or a yellow medium or a red medium, it's going to mean that we are using either repression or activation conditions in the experiments that I'll show in a minute. Okay, so I'm going to refer to an interesting project that we work together in collaboration with Brett Finlay's group, and where we decided to mutate every single gene of the pathogen ST island. And at that time, and as you can see, this study was done almost 20 years ago. Time flies. So at the time, it seemed to be like we didn't have any other thing to do. It was sort of a crazy idea, but it was actually very illustrative to do this experiment, to study in a systematic way the pathogen ST island, even when several genes were already known and the function was already known, but at least half of the island wasn't already characterized. At the time, we already know the role that this first gene play in the regulation of the island. So we pretty much knew that the first gene of the Li1 operon encoded an HNS. I'm going to be referring to these proteins. HNS is a protein that is part of the bacterial nucleus. It has to do with the structuration of the nucleation of the chromosome, but also plays a very important role in mostly negative regulation of several genes. And one of the interesting things when the sequence of the Li came out was to see that the first gene actually encoded a protein that has identity mostly in the DNA binding domain with this histone-like structuring protein that plays a very important role in repression. And it was interesting because in this case, a paralog of HNS was playing an opposite function to HNS because was in charge of activating the whole island and was in charge of actually antagonizing the function of HNS, which is the main repressor of the pathogen ST island. Just give me 1 second. I'm going to try to move the. Okay, so but one of the interesting observations of this work was to realize that one of the genes that basically had a non-function at the time had basically the same phenotype as the delta layer mutant. And what I'm showing here is a Western blot experiment of every single mutant using two antibodies against two of the proteins that are encoded within the Li, the pathogen ST island. One of them is TIR, which is this protein that is injected into a cell and then becomes the receptor of the bacteria once this protein is integrated into the cell membrane. And ESPB, which is one of the translocator proteins that has to do with the formation of a pore in the cell membrane that allows the type 2 secretion or the injectisome to actually inject other proteins into the cell cytoplasm. So basically what we are seeing here is if these two proteins in cell extracts are either expressed or not. And as you can see here, the delta layer mutant doesn't express any of the two proteins, and this, what was called ORF11 at the time, had basically the same phenotype, and the others are just control the mutant, the delta TIR mutant, the delta ESPB, which also don't have the or show the band, and well, in this case the ST mutant had a polar had an effect on TIR expression because it's actually a chaperone of TIR. So at this point, even when the sequence was already available, we didn't realize that this gene also encoded a protein that had a DNA binding domain, a typical helix-turn helix domain that had to do with DNA binding. And sure enough, its mutation also render a phenotype similar to the one that the layer mutant had. This particular gene is forming an operon with another gene that was called ORF11 at the time. And in this experiment to the right, what I'm showing is an experiment was done by overexpressing this first gene of the operon, the ORF11. And as you can see in a similar Western experiment, when we overexpress ORF11, the expression of Li proteins and then of Li genes was downregulated in a very significant manner. So we concluded from this experiment that in this operon, we had, which seemed to be a contradiction, because basically we have a gene that encoded a protein that acted as a repressor, as this experiment shows, and another one that was actually having a positive effect on the expression of the Li. And one of the interesting things, I'm not showing that experiment, is that when we generated a double layer of 11 mutant, and then we complemented with either layer or of 11 that we call now girl A, the only way to complement the expression or to reactivate the expression of the Li with those complementation experiments in the double mutant was with LAIR, which basically was telling us that LAIR was the main activator of the island, and that probably girl A was needed to activate and LAIR. And then further work after that publication came out, it started to reveal that in fact girl A was bona fide DNA binding protein that had to do with the activation of the Li1 promoter and then of LAIR, and that once LAIR was activated and the concentration of the protein increased in the cell, it basically had the function to remove HNS, which we already knew was repressing basically all the promoters of the pathogenicity island. And this also created a positive regulatory loop, because LAIR also did repress the GIRL-RA operon, and under those conditions, the increase in the expression of GIRL-A also allowed it to increase the expression of LAIR and then potentiate the whole activation. However, together with GIRL-A, we also had co-expression of GIRL-R, which later we knew, and there was also a previous paper using two hybrid system in yeast, showing that ORF11 and ORF10 interacted. And with time, the group in Singapore crystallized both proteins and actually showed something that we had an idea was happening, because based on mutations and pull down experiments, in vitro interaction experiments, that these two proteins interacted basically in the case of GIRL-A using the HTH domain, or the DNA binding domain, interacting with a pocket that the GIRL-R dimer forms, that is basically antiparallel barrel, creating this pocket where the HTH domain of GIRL-A interacts. And it was easy to suppose at the time that this interaction, and mostly because HTH was involved, that this interaction was basically eliminating the possibility that GIRL-A could interact with the LAIR promoter, and then that's why when we were overexpressing GIRL-R, basically we were titrating GIRL-A and then blocking the possibility to get the regulatory loop going, and then LAIR expression and HNS will remain as a repressor, keeping the Li downregulated. So the thing is that, as I also mentioned at the beginning, in the laboratory, we can have conditions where either the Li is induced or is repressed. And one of the questions that emerged at the time is what was happening with these two proteins. If the model at this point was pretty straightforward in terms of suggesting that once both proteins were expressed, if they interacted, GIRL-A was going to be basically inactivated by this interaction, and that was the way that the Li was going to be repressed. So it was easy to imagine at the time that this interaction was happening mostly in LB, assuming they were being expressed in LB, and that's why the Li was basically being repressed. Okay, so how LAIR acts to activate the Li promoters. So basically what I'm showing here is a what we call EMSAs, or electrophoretic mobility shift assays, where we can basically mix DNA fragments with either HNS, purified by histidine track, or the LER protein purified the same way. So what I'm showing here is a map of what would be the divergent promoter of two of the operons of the lead 2 and the lead 3, which encode basically type 3 secretion system components. And the two promoters almost overlap. And what we did in this case was just to divide this region in 3 different fragments and then use a non-related control in the same mix with the 2 proteins independently. And what we are seeing in the left panel is that as we increase the concentration of HNS, HNS binds initially to what we call fragment 2, and then that we name it silencer region sequence 2, and then later binds to a second one in fragment 1, but with less affinity, but not to fragment 3 or the control fragment. So basically, this suggested that HNS was was binding to this regulatory region, but basically flanking both promoters. And based on the characteristics of HNS, so it's easy to think that probably HNS was forming a nuclear repressor complex that was either avoiding the entrance of the RNA polymerase or trapping the RNA polymerase within the complex. Which was interesting is to see that when we did a similar experiment with LER actually bound with better affinity, slightly better affinity to fragment 1, but not to fragment 2. So which suggested that LER had binding sites near what we call the SRS1, and that was probably playing a role in displacing HNS from this region and then disrupting the complex. And we actually had also evidence using transcriptional fusions to the CAT-reported gene. This is the chloramphenicol transferase gene that we use as a reported system, where what I'm showing here is just transcriptional fusions of different fragments to the lead 2 promoter. It goes to the left. And as you can see, as we remove this, either the silencer 2 or the silencer 1, the transcription of this transcriptional fusion became basically LER independent, which strongly suggested that LER wasn't a bona fide activator, but was mostly acting as an antagonist of HNS. So in the presence of LER will displace HNS basically from this region, and I have a model of it. It's here. Basically, what was so this is a model of the nuclear repressor complex. As I was mentioning, HNS has this ability to bind to different nucleation sites, and from there either nucleate over the DNA or form this by interacting in these distant, based on the binding to these distant sites, it can form these complexes and then form these loops where either the RNA polymerase cannot access the promoter or be trapped in this complex without the possibility to start transcription. Once LER is bound to the region of where we have a silencer 1, LER displaces HNS, open up the complex, and then transcription can proceed in both directions, even if HNS is still bound to the other site. And just to move quickly to this experiment, what I'm showing here is that basically what I was saying is that based on those results and others from all the different groups, it was evident that LER wasn't needed as some other bona fide regulatory proteins, that it was basically required to displace HNS. most obvious experiment to do was to create or generate a double layer HNS mutant, but unfortunately, those kind of mutations make the bacteria very sick. And then we did in a different way. That was basically by having different mutants expressing a dominant negative version of HNS, which basically by overexpressing a mutant version of HNS that doesn't have that lost the ability to interact with the DNA, but not the capacity to form oligomers between the monomers. You can generate this dominant negative effect because by forming these heterodimers with the wild type version of HNS and the mutant, basically you can generate an HNS null phenotype. And as you can see in this, I just want to show these three different mutants where in the absence of arabinose, so we have this dominant negative version of HNS under the control of arabinose inducible promoter. In the absence of arabinose, basically we don't see expression on their repressive conditions actually in the delta layer mutant. But if we overexpress the dominant negative version of the HNS, we can complement or we can induce the expression of the Lee genes. What you are seeing here is a secretion profile. These are basically proteins that are secreted through the type 3 secretion system, and then that we can recover from the supernatant. And this is the pattern that you get in the wild type and then in the mutants. And basically what the experiment is saying, the same thing will happen in the Gurley mutant and in the double layer Gurley mutant, that by overexpressing the dominant negative version of HNS, you generate a null phenotype for HNS, and then the induction of the Lee pathogenicity island, even in the absence of layer. So was basically, this experiment was basically reinforcing the fact that layer is not a regular activator, but an antagonist of HNS, which function is basically to remove HNS from the different promoters of the Lee. And this was the model I was showing. And the other interesting thing is that later, Ilan Rosenshen's group also identified a layer binding site on the layer regulatory region, which was basically used by layer to repress its own expression. And this has the logic that once the system starts, or the pathogenicity island starts to be expressed or induced, there is a point in which the accumulation of layer also allows layer to bind its own promoter and then down-regulate its own expression to prevent that the system just goes without any control. And you can see this by just following the expression of HNS and layer by tagging the proteins in the chromosome. And as you go along the growth curve, you can see how layer eventually disappears, allowing HNS to occupy the sites on the layer and then just turning down the expression. So this is a way how the system controls itself. And so I'm gonna run out of time, but so as a summary, as I was saying, in this big complex network of different elements and that regulate the expression of Lee and that target basically the layer promoter. So we have this specific regulator, which is Gurley, that controls the expression of layer. But as I was also mentioning earlier, EPEC also contains in the EF plasmid this operon that not only encodes the activator of the BFP operon, but also a protein that has the ability to also activate layer. So this is an interesting situation from the evolutionary point of view, because these are two entities that had to do with virulence that were acquired in different evolutionary moments by horizontal transfer events, but that contain elements that were co-integrated to communicate at the transcriptional level to coordinate the expression of different virulence factors. So as the BFP operon, which is required for the initial stage of the infection, is being expressed, also PERC is being produced to give a head start to the LE, and then start this regulatory loop established between LER and GERLA, which potentiates the expression of LER, and then the LE, which then proceeds not only to the localized, to perform localized adherence, but also intimate adherence through the expression of the type 3 secretion system. PERC is a very interesting protein because it doesn't have homology to anything, doesn't have homology to, or no, it doesn't have DNA binding motifs. So we still understudy to understand how actually has this similar function than GERLA, but acting differently because basically we still don't know how it actually activates expression of LER. But even in the absence of GERLA, overexpression of PERC can nicely activate expression of LER and then the LE. I'm not going to go into the details of how GERLA works because I hope to have the opportunity to talk about it in another opportunity. But GERLA is also an interesting protein because based on our studies and studies from Steve Busby's laboratory, basically established that GERLA binds in the spacer region between the -10 and the -35 boxes of the LER promoter, and compensating the fact that instead of having 17 base pairs between the two boxes, it has 18. That was the proposal that basically is correcting the difference in the distance to allow the RNA polymerase to start transcription, although we believe that something else is happening because this particular promoter region has other features that have to do with the control of LER expression. But the other interesting thing in terms of how these things are co-integrated and used by the cell to control other things is that GERLA and these particular features of the promoter are conserved in a few of the effector genes that are encoded outside the pathogenicity island. And just to give you an example, these are 3 genes that encode proteins that are completely different between them, but that actually share the regulatory region, including an inverted repeat that we believe is also the target of another regulator. But GERLA, in a way, is also coordinating the expression of some of the effector proteins that are encoded mostly in prophages, in IPEC and IHEC and Citrobacter. So just to quickly show you some other results regarding a GERLAR, the repressor. As I was mentioning, if we knock out HNS, if we eliminate HNS, the LE genes are efficiently expressed in the absence of that repressor, either under repressing conditions. But we knock out GERLAR, the same thing happens. So basically, we get the same phenotype when we mutate GERLAR and grow the bacteria under repressing conditions. So it seems that based on these results, it was obvious that both GERLAR and HNS as an ancestral regulator in IPEC, and GERLAR acquired together with the island, had a strong repressor effect on the pathogenicity island. But what was interesting is when we decided to, and as I was mentioning before, so basically the model was that GERLAR, by interacting with GERLA, prevented the activation of LER, and that was the way GERLAR was repressing, which in fact happens. But when we did this experiment, basically the hypothesis was that in the absence of HNS, LAIR wasn't going to be required, and then GERLAR shouldn't repress because the system was basically constitutively on, and in the absence of HNS. But what was striking is that by expressing GERLAR in the HNS mutant, we still got repression of all the pathogenicity island, but not of other genes in the chromosome. So it wasn't an artifact of the experiment. It was something that was specifically happened on the lead genes. And then to make a long story short, other experiment that gave us a hint of what was happening in terms of the function of the heterotrimer that was formed by the dimer, the GERLAR dimer and the GERLA monomer, which we thought basically inactivated GERLA. We did an experiment taking advantage of GERLA mutants that were inactive in terms of activating LAIR. So they are basically defective in DNA binding. But in one case, this R54 mutant was still able to interact with GERLAR, but not I44A. So this mutant was inactive as a DNA binding protein, as an activator, but also as a protein that interacts with GERLAR. So this is a pull-down experiment, and these are secretion profiles. So when we overexpress together with GERLAR, either mutant, we got a very interesting phenotype under inducing conditions in the wild type strain. So if we overexpress, that was the experiment that I showed earlier, overexpression of GERLAR basically repress the expression of all the lead promoters. So when we overexpress this mutant that doesn't longer interact with GERLAR, we got the same repression phenotype. But when we did it with the mutant that still interacts with GERLAR but cannot activate LAIR, basically we prevented repression, which in other words was suggesting that the formation of the complex, the GERLA complex, was also playing a role in preventing that GERLAR repress in this independent repression pathway or activity that didn't have to do with interaction itself, but interaction in this case was acting as an antagonist activity. So GERLA was also an antagonist of GERLAR in this situation. So basically, the formation of the complex eliminated the function of both of them. So the bottom line of all this, the message is that both HNS and GERLAR indirectly and in a cooperative way act as repressors of the lead. So by mutating any of the two, we basically got the same phenotype, constitutive expression of the lead, mostly under repressing conditions. And this can easily be explained. I'm going to move to this figure because and perhaps it's easier to show it here. So basically what happens when we knock out GERLAR is that we have enough GERLA free to activate LAIR, and then LAIR basically generate or activate, displaces HNS from all the promoters and activates all the lead genes. So basically by mutating GERLAR, we are sort of generating a double mutant because there's going to be enough LAIR activated by the free GERLA to remove all the HNS. When we knock out HNS, in general, what we thought is happening is that when HNS is going to release the expression of all the promoters, so LAIR is not going to be needed, but still LAIR is going to be there to activate both GERLAR and GERLA, and then having enough GERLA to prevent GERLAR from repressing through this pathway that we still have to understand. So we know that this is a strong repressor, even in the absence of HNS, that is also specific on Li genes, that doesn't have to do with interaction with GERLA. So one of the questions for the future is trying to understand how GERLAR is actually repressing in a way independent of the interaction with GERLA, and also trying to understand how the growth conditions control the levels of both proteins and actually control the interaction to allow either to activate the system or repress the system. Because quickly, I'm just going to show this experiment, which is a native page experiment where we have either GERLA tagged with flag or GERLAR tagged with flag. And the experiment, what it's telling us is that both proteins are expressed under both growth conditions, repressive conditions and inducing conditions. And then what we think, or what we actually is our interest to find out, is how both conditions control the interaction between the two proteins or the stability of the complex, and also the relative levels of GERLAR versus GERLA depending on the growth conditions, and how those changes have to do with either induction or the repression of the system. As you can see here, these experiments are interesting because we can easily see the formation either in this case of the dimer and the GERLAR monomer. And when GERLA is present, we can actually see GERLA in the gel because it's forming a complex with GERLAR. Otherwise, GERLA doesn't go into the gel, and so this is an experiment, an easy way to actually look at the interaction under different conditions. But we thought we were going to be seeing something more striking that will allow us to understand what was happening under both growth conditions, but it just made it more interesting because basically the proteins are present independently of the activation state of the Li, but obviously had to do on how these proteins are interacting in response to the growth conditions. So just to give you a few quick conclusions. So there is the master regulator of the Li acting as an antagonist of HNS. GERLAR represses Li gene expression by interacting with GERLA and independently of this interaction in a way that we still need to elucidate. And also an interesting finding of these recent studies was that GERLA has a dual role as a positive regulator, not only acting as a bona fide regulatory protein binding to DNA, but also by acting as an antagonist of GERLAR by forming these heterotrimers. And as I was mentioning, some of the current questions and future research will be to define how the growth conditions influence the interaction between GERLAR and GERLA, and how actually GERLAR is repressing independently and specifically the Li promoters. So I just would like to acknowledge current and past members, and not all past members are in this slide, but I basically quickly took you through 20 years of history in the lab, and it's interesting to see that we are still a lot to learn, and I just can appreciate and be grateful for all the people that have worked in the lab to help us to understand what's happening with the regulation of the Li. Well, thank you very much, and
thank you, Jose, for the very nice journey into your research, and I really enjoyed listening to you. So I have a couple of questions while we wait for others to chime in. So I thought, I mean, it's been a while since I attended the pathogenesis talks at Cold Spring Harbor. I thought HNS was part of the core genome, right? But you were telling me, isn't HNS part of the Lee operon?
No. Sorry if I misled in the explanation. But HNS is part of the core.
Right.
The core genome is one of the ancestral, of what we call ancestral regulator.
Okay.
And
So
global
that,
regulator at the same time.
so
So
that's
HNS is actually interesting because it was considered the evolution sentinel by other groups. And basically what is being proposed is that when bacteria acquire something by new genes, by horizontal transfer events, HNS is one of the proteins that actually act to control the expression or to the out-of-control expression of that new information until the bacteria learn how to control it in its benefit. So, yeah.
wow, very nice. So clearly, I've been missing that part of the journey in the transcription world. Dr. Flack, chime in anytime. So go ahead.
Okay. Can I ask kind of in terms of the reservoir, what is known much about the kind of wildlife? And so is it that it came to recognition because we were domesticating cattle, and where was it hiding before?
So it's interesting. The case of IHEC cattle is the reservoir. So IHEC can live in cattle without causing disease. So actually the first major outbreak of IHEC, when actually O157H7 came to the scene and became famous, is because it actually infected several people, around 200 people, by eating undercooked hamburgers. So yeah, one of the main sources of infections is meat. Although after those outbreaks, it started to generate outbreaks also from produce. So yeah, it's a big problem in that regard. And for the case of IPEC, sorry, there is not known, as far as I know, an environmental reservoir. So it's basically transmission from humans, babies that are sick.
And in terms of the virulence, is it kind of cumulative? So is the hypothesis that the more pathogens in the islands you have, the more virulent you are? Is that, or is it not as simple as that?
I will say that it's not as simple as that. In the case of IHEC and IPEC have, you compare IHEC and IPEC with other pathotypes of E. coli, or even with commensal E. coli. These pathotypes have around 1,500 genes that are not present in other pathotypes or in commensal E. coli. So not all of them have to do with virulence, as far as we know. Perhaps under certain circumstances, some of those genes are going to have a function. But in general, the Li is considered the most important virulence factor for this family of pathogens, because if you knock it out, they become completely attenuated. Although there are other genes that also encode things or proteins that are important during the colonization process. To answer your question, so it's hard to know if that is going to mean more virulence, because if we also mention as an example Salmonella has two very important pathogens, the islands. And one of them is very important to allow Salmonella to be internalized by non-phagocytic cells, while the other is very important to become an intracellular pathogen, to be able to replicate inside macrophages, for example. So yeah, you can say that it's more virulent in that regard, but it's just that it depends on the niche where Salmonella is at the time. And I would say that it's probably the same thing for IPEC and IHEC and any other pathogen.
Sure.
I totally agree. It's, clearly the more virulent the strain appears to have acquired, especially when it comes to resistance, right? AMR, they appear to acquire more and more islands through horizontal gene transfer that allows them to survive. And I always say, not only, we identify genes and give it a name, but these ancestral genes, that means they must have had a function, and they must have a function in their bacterial survival, in their niche environment. And some of these elements, when the pathogen crosses over to human, become virulent. So it's evolutionary. I think acquiring genes is bacterial's based evolutionary survival in any environment, and then some of them becomes virulent or becomes contribute to the pathogenesis during infection.
Yeah. I think I will say both, right? Some of them are gonna give the bacteria advantages to colonize environmental niches. Some others are gonna give advantages to colonize a particular host. And it's interesting because what you're saying has to do with what I was saying at the beginning, that when we study gene regulation, for example, we create conditions in the lab, but that's probably going to be really far from what is happening in the intestine, where the bacteria are actually receiving way too many signals. And those signals are actually continuously changing during the transit in the intestinal tract. So and even when they reach a particular niche, because then there's the microbiota, then is the how, the food that that particular host consumes, etc., etc. So it's interesting because we never know. And just to give you another example, IHEC has expresses a fimbria that is expressed at lower temperatures than 37. So, at the beginning, you always think that probably has to do with the colonization of a host, but actually that particular fimbria is very important for IHEC to colonize, grow and resist treatment of lettuce or spinach leaves with chloride. So because it can hide in the stomata of the leaves and then resist treatment of this fresh produce, and since IHEC has a very low infectious dose, so that's why lettuce and spinach can cause serious outbreaks.
So they shouldn't be eating salad. Cook your food.
But or wash it. Yeah.
Oh, wash. But stomata, you're not going to wash away from the stomata, right?
Well, some of them actually resist quite a bit of chloride treatment in the stomata. So they hide there and then they survive, and you just need a few bacteria to cause an infection.
Yeah.
In the case of
yeah, I want to take you back. So what sigma factor does they use to transcribe all these operons? The Lee operon. Sigma. Also, it's a housekeeping sigma factor.
Yeah.
Okay, so because the way
and the Lee is basically expressed in the exponential phase. So you recall this experiment where I was showing expression of LAIR and HNS along the growth curve. Basically, you see LAIR during the exponential phase, and it starts to fall down when it's entering a stationary phase. It's a really nice control, because at that point you don't have LAIR, and then since HNS is constitutive, it quickly occupies the Lee promoters to turn them down.
So if that's the case, there must be a very strong regulation through proteolysis of the proteins.
Yeah.
So
LAIR is quite unstable in comparison to HNS. But
So
while
that
this loop is working, you have LER all the time until LER represses its own promoter.
So that leads me to think that some of your regulators could be proteases. And have you looked at any of those players have a proteolytic activity?
Yeah, actually, Gerlar is controlled. So it's interesting because it depends on the pathotype you refer to. In IHEC, Gerlar is controlled by proteolysis. So Gerlar is, in IHEC, the allele is preferentially expressed in the stationary phase. And Gerlar is degraded in the stationary phase. But that doesn't seem to happen in IPEC.
Okay,
So there are also
interesting.
differences, and I think it might have to do with the niche that IPEC and IHEC colonize, because IHEC preferentially colonizes the colon, while IPEC prefers the small intestine, and the signals that they're going to find are quite different.
So here's another question: have you ever looked to see if some of these proteins, like GERLR and GERLA, where are they localized? Don't assume they're localized in cytoplasm, right? Because I don't believe that.
Well, there is an interesting paper a few years ago where they actually show that GERLA is bound to the membrane.
Yes.
And the shear force when IHEC, this is IHEC again, when the shear force of the interaction with the cell surface removes GERLA from the membrane and then goes to activate LER expression. So that's the model in this paper. It's a very interesting paper, and we are not sure it happens in IPEC, but
I think you have to do the experiment. There's no guarantees.
Yeah,
right.
But there is strong evidence in that work. So
I mean, it's in fact, when I started working in Pseudomonas, nobody believed me when I said the regulator is a membrane protein, right? Because I was just gazing at the sequence and gazing at the hydrophobicity of the stretch of sequence, and I said this got to be a membrane protein, right? And now we know the regulated intermembrane proteolysis controls the whole thing, and so many regulators, and they were all part of a single operon, quite a few of them part of the single operon. So we had the 3 proteins in the membrane, and one of them was controlling the master regulator, which turned out to be a sigma factor, right? But I think the where I started was just plotting hydrophobicity plot for all the proteins that are involved in the system. And then once you have an idea of where they are localized based on sequence, then we went and did the experiment to show where they're localized. It looks like for me, even a GERLR could be sitting on a membrane. And Steve Busby should have done that, because he wrote a fantastic review many years ago arguing that it's only the tip of the iceberg that we're going to find more and more regulators on the membrane. It's
a good point. In the same work, they didn't see GERLR associated with the membrane. They saw GERLA, but not GERLR. So it's interesting. But I agree with you that I think that the subcellular localization of regulators plays a very important role in the way they work.
No, so especially when your system, you're saying that you have the medium that can control one versus the other, then which means that the regulatory system has to recognize a signal from outside the cell. Right? Yeah. So what is the signal? So that means that you must have something on the outer membrane that transfers the signal to a periplasmic component, which then transfers the signal to inner membrane and then into it. So do you have any 2 component systems involved in the process?
Yeah, this first slide that I show with all these elements, there are some two component systems, and it's interesting because they respond to different signals. So the integration of all these things, have a role in modulating the expression of the Li, and in particular of the specific regulators which are essential for Li transcription, which will be GERLA and LER. So, but the modulation of LER through all these different regulators, it's actually allowing the bacteria to respond to under different circumstances. And there now is known that several small RNAs also play a role in modulating specifically not only LER but also other promoters or other genes, even if they're internal to some of the operants. So there's also play, in coordinating the expression, because probably not everything is needed at the same time. So even one of the chaperone of TEER, says T, interacts with CSRA, which has to do with messenger control. So that this protein controls the translation of different transcripts, and you wouldn't expect that a chaperone of encoded in a pathogenicity island that actually is a chaperone of an effector protein will interact with a protein that has a very important role in controlling different things in the cell. So the fascinating thing of how the fact of how these things evolve is that how many things come into play and get integrated even when they didn't exist together in the ancestors. So
yeah, very nice.
It just becomes more complex.
Yeah, it gives you a lot of time for a lot of research. Norman, do you have any more questions?
I don't know if you know, Jose, but I'm interested in the treatment of, so in STEC, so is it right that you kind of avoid antimicrobial treatment because you don't want to release the toxin? So are people looking at kind of anti-toxin treatments?
Yes, there are actually antibiotics that you can use, because there was a Japanese group that actually did a very interesting study comparing the effect of different antibiotics, and that's also a fascinating phenomena, because some of the antibiotics actually generate this stress signal that makes the STX phase enters like the lysogenic phase. And then there is just a circle, because the STX genes are under the control of late promoters. So when the phage starts to become lysogenic, it's not only that the STX gene is more expressed, but also the fact that the phage is being assembled just lyse the cells, and then more toxin is released. So it's very dangerous in some cases to treat with certain antibiotics. I don't remember the list of those that you should not use, but some others actually still can be used without causing this effect. But there are groups studying other strategies to control EHEC problem, because the thing is that it can be deadly or cause this kidney failure that can be not reversible. So
very nice. All right. Okay, thanks. So let's wrap up for the day. This has been an amazing hour and a half of excellent science and discussion, and I thoroughly enjoyed your presentation, Dr. Puente. And I really like to thank Dr. Fry, who joined us today, and I hope he'll come for every session. And I thank the staff, particularly Dalia and Akaidon, for helping us to set up the lecture today. And I'd like to thank once again the audience. Please, a reminder that a link to the recording will be emailed to you, and it'll be available for you to view for yourself or with your friends for 7 days. The next session of JMM series will be in April 28th. We haven't announced the speaker yet, so please keep a lookout. Check our website, but I will certainly hear from us. But however, we don't have any talk for March because the Microbiology Society annual conference is in March, and I hope to see many of you there. And please come and look for us. Come to the booth where usually we hang out and connect with us. With that, thank you all and have a great weekend, and see you all soon in person.
Thank you, Claire. Thank you, Norman.
Thanks, Jose. Thanks.
Thank you, Dale. Bye-bye.
Okay, bye.